Hi everyone, I am Siti Rainisa M. Haji Sirad together with my partners Makarambon Umilhaya, Bimbantas Najmiya will discuss to you the chapter 35 of the Bailey Scott book, the Brucella. Did you know that the family of Brucelliaceae comprises three genera, the Ochrobacterium, Mycoplana, and Brucella? Together with my partner, we are going to discuss the Brucella in this chapter. So let's move to the general characteristic of this Brucella. Brucella are small, facultative, which means it can survive both inside and outside of the cell, non-motile and intracellular. It is also an aerobic, which means it needs oxygen to survive. It is also a gram-negative cocobacilla or short bacilla that stain poorly by conventional gram stem. Many isolates require supplementary carbon dioxide for growth, especially on primary isolation. Brucella are closely related to Bartonella and Agrobacterium. Next slide, please. So the next slide is the epidemiology and pathogenesis of the Brucella. The disease of brucellosis occurs worldwide, especially in Mediterranean and Persian Gulf countries. These organisms are capable of survival for extended period, which is in soil, 10 weeks, in aborted fetuses, 11 weeks, bovine stool 17 weeks, and milk ice cream is 3 weeks. They can survive in freeze cheese for several months. Brucellosis is a zoonosis and is recognized as a cause of devastating economic loss among domestic livestock. Humans become infected by four primary routes. First, ingestion of infected unpasteurized animal milk products, which is a most commonly means of transmission. Second one, inhalation of infected aerosolized particles. The laboratory acquired infection is the most important source of transmission. Third one is the direct contact with infected animal parts through ruptures of skin and mucous membrane. And the last one is the accidental inoculation of mucous membranes by aerosolization. And the next slide is the respective and natural animal host. The first one is the Brucella abortus, which can transmit by cattle and buffaloes. The Brucella militensis, which is the sheep, goats, or camels. Brucella suis can be transmitted by swine and a variety of wild animals. Brucella canis can be transmitted by dogs. Brucella ovis can be transmitted by rams, which is not associated with human infection. And the last organism of this Brucella is the Brucella neotome, which is transmitted by desert and wood rats, which is not also associated with human infection. In the next slide, we are going to discuss the spectrum of these diseases. The clinical manifestation of brucellosis vary greatly, ranging from asymptomatic infection to serious debilitating disease. For the most part, brucellosis is a system infection that can involve any organ of the body. So the symptoms, which are non-specific, include fever, chills, weight loss, night sweats, headache, muscles aches, fatigue, and depression. Lympan- Lymphadenopathy and splenomegaly are common physical findings after an incubation period of about 2 to 4 weeks. The onset of disease is commonly insidious. Complications can occur such as arthritis, inflammation of the vertebrae, genital, pulmonary, and renal complications, and endocarditis. Relapse is considered an important feature of brucellosis. It is associated with delayed initiation of treatment, ineffective antibiotic therapy, and positive blood culture findings during the initial presentation. And the next slide will be presented by Omilhaya Makarambon.
So let us now proceed on the laboratory diagnosis of the brucella. First is we have the specimen and collection of transport and processing. So samples for the diagnosis and identification of infections associated with Brucella species may be used for culture serology and or nucleic acid based testing. And we have the gold standard for the definitive diagnosis of brucellosis is isolation of the organisms in cultures of blood, bone marrow, cerebrospinal fluid, and pleural and synovial fluids, urine, abscesses, or other tissues. If processing will be delayed, the specimen may be held in the refrigerator. Some research laboratories offer nucleic acid-based testing for the identification of the organism. We have here the essential that the clinical microbiology laboratory be notified whenever brucellosis is suspected. First one is we have the to ensure the specimens are cultivated in an appropriate manner for optimal recovery from clinical specimens. And the second one is to prevent accidental exposure of laboratory personnel handling the specimens because Brucella species are considered category B select agents, which is a specimen level should indicate that Brucella species are potential pathogen. And let us continue to the direct detection methods. So direct stains of clinical specimens are not particularly useful for the diagnosis of brucellosis. And um, the sensitivity varies among assays ranging from 50% to 100%. First one is we have the conventional and real-time polymerase chain reaction known as the PCR assays. They are reliable and specific means of directly detecting Brucella organisms in clinical specimens. And the next one is the MALDI Biotyper software. It is a convenient molecular method that can be used for diagnosing brucellosis. This molecular method requires strict culture conditions and sample preparation. And for the cultivation, although most isolates of Brucella species grow on blood and chocolate agars, some isolates are also able to grow on Makoni agar. So, Brucella agar infusion base is recommended for specimen types other than blood. Culture should be incubated in 5% to 10% carbon dioxide in a humidified atmosphere. As we can see on the figure 35.1, we have the growth of Brucella species on chocolate agar after incubation of for 2 days. We have here the letter A, and for the 4 days is the letter B. So next is the approach to to identification. Brucellosis is the most commonly reported laboratory-acquired bacterial infection. Therefore, all handling and manipulations of suspected Brucella species should be performed in a class 2 or higher biologic safety cabinet. Gram stain of the organisms reveals small cocoa bacilli and resembles fine grains of sand. So first is uh, Brucella species are catalase and urease positive, and most strains are oxidase positive. Other non-fermentive gram-negative bacilli that may be confused with Brucellae are Bordetella, Moraxella, Kingella, and Acinetobacter species. Brucella species, however, are non-motile, as Sarah earlier mentioned, urease and nitrate positive, and strictly aerobic. Brucella species are differentiated by the rapidly with which the organism hydrolyzes urea, its relative ability to produce hydrogen sulfide, and its requirements for carbon dioxide is susceptibility to the aniline dyes, threonine, and basic fusion, which shows in 35.2. We have here the Brucella Brucella militensis with traditional gram stain. This is the letter A, the Brucella militensis with traditional gram stain. And for the letter B, is a gram stain with 2 minutes of running counter stain to allow easier visualization of an organism. So um, this, as we can see on the table, characteristic of human pathogenic Brucella species. We have here the species with which is the Brucella abortus, Brucella militensis, and Brucella suis, and Brucella canis. The carbon dioxide required for growth for the Brucella abortus, it is positive or negative. For the Brucella militensis is negative along with the Brucella suis and canis. The time positive for urease is for Brucella and Brucella abortus and Brucella militensis are 2 hour and rare 24 hour. Same with the Brucella suis and canis, they are 15 minutes time to positive urease. And the hydrogen sulfide produced for the Brucella, it is positive most strains, and for Brucella militensis is negative, and along with the Brucella canis. And for the Brucella suis, it is positive or negative. So as we can see here, the inhibition by dye, we have the threonine and fuchsin. And now we will going to proceed to Najmia.
Thank you so much to the previous reporter, and now we're going to jump on to the last one, or my part, which is the sero diagnosis. I'm going to give a brief and precise discussion for this one. So we have the serologic test is widely used, such as the serum agglutination test or microplate agglutination. This technique detects the antibodies to Brucella abortus, militensis, and suis. For the Brucella infection, it follows the classic antibody response, which is the immunoglobulin M appears initially and followed by immunoglobulin G. So basically for this one, we have in the immunoglobulin M appears first and then followed by immunoglobulin G. That is the antibody for the Brucella infection. So the SAT can cross-react with the class immunoglobulins with a variety of bacteria. So it cross react with glass immunoglobulin such as the classic antibody response wherein immunoglobulin M appears initially before the bago yung immunoglobulin G. And then we have here the ELISA or the enzyme linked immunosorbent assay, and it has been developed and it uses purified lipopolysaccharide or a protein extract. Next slide, please. For the additional serologic assays, we have the lateral flow dipstick and also the immunocapture agglutination method. For the immunocapture assay, it demonstrates sensitivity and specificity similar to a COM test, and it comes test and it less cumbersome to perform. So on the other hand, the dipstick test has a high degree of sensitivity for about greater than 90%. The microarrays that contain numerous antigens are being evaluated for their efficacy in elucidating differential antibody responses from patients during different stages of the disease. Um, so here we have the dipstick. It has the higher degree of sensitivity, which is 90%. And then mentioned in these microarrays, it contains numerous antigens, and for the different antibody responses from patients during different stages of the disease. So next slide. So Brucella is not reliable in vitro susceptibility testing because of its fastidious nature and intracellular localization. To prevent relapse of infection, so yung mga patients with brucellosis, they must undergo a prolonged treatment for like 6 weeks. Not compared to other infection, they could be for like 3 weeks, 2 weeks, but for brucellosis, it must undergo prolonged treatment for 6 weeks with antimicrobials that can penetrate macrophages and act in the acidic intracellular environment. And for the initial therapy for this one, we use, the most recommended one is the deoxycycline or the tetracycline in combination with the streptozyme or rifampin, and which is highly recommended for the initial therapy of this antimicrobial susceptibility testing and therapy. Next slide. So for the prevention, Brucella infection have been developed. The successful vaccine against Brucella infection have been developed for livestock. So so if we're going to say the most effective to prevent this one is the development of the vaccines, because it has been developed to the livestock, and the development of human vaccine has met with the serious medical contraindications and low efficacy. The prevention of brucellosis in humans depends on the elimination of disease in domestic livestock. So for this one, same with our situation with COVID-19, vaccine is very successful, and we are encouraging to get everyone vaccinated. And that is the end of my part, and that is the end of chapter 35. This is Najmia Mimbantas, and this first reporter was Miss Rainis Haji Sirad, and the second reporter was Miss Omilhaya Makarambo. Thank you so much for listening.
Thank you, and that's all.